Seed train is quite a simple process. You have to expand the cell from a working cell bank to that you start usually at 30 milliliter up to a large-scale production bioreactor that can be between 1,000 up to 20,000 liter. So at each step, the cells are transferred from one step to the other. The key reason to do that is to keep the cells in their logarithmic growing phase.
Welcome to the Smart Biotech Scientist podcast. I'm your host, David Brohlman, and today we're diving into a critical yet often neglected aspect of biologics manufacturing, the seed train stage. I'm thrilled to have with us François Carruzzo, who is the co-founder and CTO of Bioscibex, a Swiss startup revolutionizing cell expansion technology. From managing multiple vessels to preventing contamination, François will share insights from his journey into transforming traditional seed train approaches. Whether you're sipping your morning coffee or commuting home, get ready for some game-changing perspectives on bioprocess development. Let's jump right in. Are you juggling the complexities of CMC development while trying to enjoy the beauty of biotech? Have you ever wondered if there's a way to simplify bioprocessing? Welcome to the Smart Biotech Scientist podcast, where we're diving headfirst into the very challenges you face. We're breaking it down, demystifying the jargon, and giving you the keys to unlock your full potential. I'm your host, David Brohlman, and I get it. With 15 plus years in the biotech industry, I face the same challenges you do. There's a way to simplify and streamline so you can remove complexity, you can skip trials and errors, deliver without delay your groundbreaking therapy to clinics and market, and still enjoy every single step. Do you want to learn how industry experts and I did it? Grab a cup of coffee and your favorite notebook and pen. Now is the time to take your bioprocessing game to the next level. Let's smarten up biotech. Welcome, François. It's so good to have you on today.
Thank you very much, David, for inviting me for this conversation.
Yeah, I was looking forward to this conversation because we have talked about it for quite a while, and now finally we have the time and the opportunity to do this interview. So, François, I would love to have your take on this question I usually ask at the beginning of my interviews. Share something that you believe about bioprocess development that most people disagree with.
I think a lot of people in the industry think that cell expansion requires a lot of multiple vessel steps to remove unwanted elements. In fact, from my experience, passaging cell is simply diluting cells and not really eliminating something. So you eliminate by just dilution.
Before we dive deeper into the world of cell expansion and also the passion you have for this area, draw us into your story, because your experience covers everything from E. coli at Berkeley to mammalian cells at Merck before co-founding your own company. What sparked your interest in bioprocessing, and what key insights from your journeys shaped your vision for transforming cell culture, and what were some interesting pit stops along the way?
So I've always been fascinated by fermentation. Even at some point, I was brewing my own beer in my kitchen. I think on the other side, my dad worked in the big industrial company. So I was really working in a large-scale machinery environment. That led me to EPFL and ETH when I finally find my way in biopharma industry. More about funding the company. I've seen a lot of inefficiencies in the industries, especially for the cell expansion. So I really like to find a way to simplify and make it streamlined. That's what drives me for the development of Bioscibex.
Fantastic. So let's start with the beginning. Could you explain what a seed train is and why cell expansion is such a critical part of biologics manufacturing?
Seed train is quite a simple process. You have to expand the cell from a working cell bank that you start usually at 30 milliliter up to a large scale production bioreactor that can be between 1,000 up to 20,000 liter. So at each step, the cells are transferred from one step to the other. The key reason to do that is to keep the cells in their logarithmic growing phase. So usually it means moving from a density of 0.2 million cells up to 4, 5 million cells per ml. So it's really critical to keep the cells at high density, high viability, and also makes as much as possible to reduce the overall production time. But I think in general, this step is not really the big attention for biopharma. There is more energy on the final production bioreactor because that's where the product quality comes. Because during the cell expansion, the cells are not really producing that much yet. But I think in the future, biopharma will have to streamline their process and improve it. So they will also focus on the cell expansion.
Yeah, that's right. I mean, you hear all the time about productivity, about the production stage, and we actually forget what's happening upstream of that. And depending on the scale you're finally producing, if we talk several thousand liters, actually your seed train is quite long. Because for those who are not familiar, basically the seed train starts when you thaw your vial, where you have a very small volume, and then you need to increase the volume to have enough cells for your production bioreactor. So this can be quite long, right? What are typical time or durations for that phase, François?
Yeah, so typically the duration up to 2 weeks, and usually the production is 2 weeks. So it's half of the time.
And what are the traditional stages, or like what is typically being used in the industry to go from the vial finally to end up in the production bioreactor?
So usually for now, everyone is using spin tube, shake flask. Then for really small volume, 40 milliliter up to 1 to 5 liters. And then they move to wave system, usually the rocker motion system. There is a lot of alternative there. Then it moves to the stir tank bioreactor, starting from 50 and up to the final product, so in the big bioreactor.
And how have these technologies changed over the last 10, 20 years? Or do we use pretty much what we have actually used 20 years ago? Or what are the recent innovations in that space?
I will say, yeah, it has really evolved from glass on a reusable system to the single-use system. So I will say that's the main move. The wave system was kind of the first alternative to like spinner or glass vessels. Now, I mean, everything is single-use. So the shake flask, then the wave or rocker motion bioreactor, and finally the stir tank as well. Everything is single-use. So I will say that's the main factor. Then also big attention was put in the N-1 with a perfusion system. So the goal is kind of reducing all the expansion step from 30 milliliter to the N-1. So N-1 was intensified, so you can reduce its size. And on the other side, cell bank with higher cell density was developed. So you can start, instead of starting at 10 ml or 30 ml, you can start at 60, 100 ml. So typically the big innovation.
Can you just explain what N-1 means, or N, what does it stand for?
So N is the last final step, the production bioreactor, and the N-1 is the step before the production bioreactor, usually the last step of the cell expansion.
Now, well, you're right. We talk a lot about the production reactor, and we also talk about quite a lot about the N-1, especially when it comes to an intensified one or perfusion or whatever. There's a lot that has been done in that space. But now we should not forget that cell expansion is a critical stage for the entire fed batch, or if you go into perfusion and afterwards at the production stage, it's critical. So can you just highlight what are the challenges scientists and companies are facing today to make this reliable, to make it cost effective, to reduce risk, to be consistent, and so on?
Today, all those steps make it complex to handle. So you have to have a precise scheduling and take into account the variability of the process. The goal is to avoid any missed production windows. Because if your cell expansion doesn't work, you will maybe miss production slot or lose the media that is ready in the production bioreactor. And That's one side, so it's the organizational. As the first step are still, there is a lot of step, and also the first step are open step, there is a lot of risk with the contamination. So everything is performed in open vessels, so there is always a risk of contamination. And same thing, if it contaminates, the risk is you lose the whole slot. Also, reorganizing, and then you have to deal with the deviation process, can be a big issue.
So what I'm hearing, Francois, it's because it's open, you have quite a high contamination risk. And what I'm also hearing that it's quite manual in certain cases. I mean, especially if you are at the smaller sizes like the spin tubes or the shake flasks.
Yeah, every 2 days you have to sample, analyze, then dilute. There is some calculation, quite easy calculation in between, but during all those steps, you have to open a risk of contamination. The goal is really to keep everything closed, and when it's closed, the risk of contamination is really, really low. And also, there is all the risk with the wrong sampling, the managing of the sample that can lead to wrong value, and then you delete too much your cells, and then you're outside your spec. It will lead to a longer cell expansion or even maybe stop of the cell expansion. So that's typically how it's not really complex, but as it's a lot of steps, there is the risk that one fail is high.
Speaking of complexity, certain companies have multiple products running at the same time, multiple campaigns. So there's also this operational complexity, right? Different media, sometimes even different containers. From my experience, we have processes that were using shake flask, other spin tubes, and then others they had the stirrers, some legacy products and so on. So I think there's also a lot of operational challenges coming from the quite manual still handling of all that.
Yes, all the product might end up in the same biosafety cabinet. So it means that each time you have to ensure you clean it and you don't have any cross-contamination that is possible. So typically, even if you can use the same room on the same incubator for several products, you will have to really make sure you're not doing any contamination, and that leads to a lot of work on a lot of organizational work on cleaning. That typically, if you have completely separate process and you don't use the same equipment for multiple process, it's easier. You could also have multiple room instead, but it will be really, really expensive. So not ideal.
As you have said, François, a bit earlier, so there have been some innovations. For instance, we have now wave bags and these kind of solutions. If we look at the industry today, what technological limitations have prevented or are still preventing innovation in the cell expansion space?
So I will say for the cell bank, it's more about moving from something which is really simple and works to something that you have to, because the cell bank will be the same for the whole product lifetime. So I think people prefer to stay conservative there. On the other element that brings advantage is the turn down ratio, so the volume range of the reactor. Our solution has a huge turn down ratio, but also competitors have developed now bioreactor with larger turn down ratio. So you can decrease the number of steps.
So tell us a bit more about how you have developed a single-use bioreactor. So you're the co-founder and CTO at Bioscibex, and you are developing a solution that changes the game in the cell expansion and especially addresses all these limitations we have just talked about. So tell us a bit more. What is your system? How will that change the whole cell expansion? So make us dream a bit.
The solution is really simple. It's an evolution of the rocking motion bioreactor. It allows to control over a much wider volume range from 30 milliliter to 30 liter in the same single vessel. So you can do all your steps continuously from 30 milliliter to 30 liter. You start with your vial towing, you insert it inside our system, and then you continuously dilute your cells up to 30 liter. And if you want to go higher, we will develop perfusion system, compatibility with perfusion system. So you can even go higher in cell density. So the goal will be you suppress all the steps you do. You just have one bioreactor from the vial towing up to the production bioreactor. The only bioreactor you need is the production one. Typically, our competition is doing a turn-down ratio, so the volume range of 5 to 10 in the same bioreactor. In our case, we are targeting 1,000, so a factor of 1,000. That's this innovation that allows us to do the continuous cell expansion.
So that means that you will have one single container from the small volume until ideally when you're inoculating your production bioreactor. Obviously, there's limitations as you highlighted. Currently, it's 30 liters, but let's say you need 30 liters. You can now do all these open steps and complex handling actually in one single system, right?
Yeah, correct. The advantage is that this system is completely closed, so you can move it in the bioreactor room. So you don't need to have a high classification room for the manual step at the beginning. So it will also reduce your space for that.
And what are the advantages, obviously, besides the obvious one, you reduce contamination? How does this, for instance, impact the robustness of or the variability of your cell expansion?
So typically, we can mimic exactly what happened in the shake flask. I think it's not the ideal solution because the goal is really to have a continuous dilution. You don't want to move from minus 20 to 30 degree in really minute, in second. It's ideally we want to have a smooth transition. So the continuous dilution is way better than having every time the cells back to the phase. So you want to keep them in the optimal dilution.
And I imagine you can do that even better than now currently with the traditional system where you're in a batch mode. I think using your system, you could do that in a continuous mode, right?
Yes. So really the vision of the system is to be able to do a continuous cell expansion. You can optimize your doubling time because you have optimal dilution for your cells. So they are exactly at the dilution they prefer. Then you can wait up to the maximum volume and then it goes higher in intention. So typically you start at 30 milliliter, they grow up to 1-2 million cells per ml. And finally, when they reach 30 liter, you have to let them grow higher in cell density. And then you can install a perfusion system if you want.
And you could even do this if you then start a fed batch, or is that problematic because you train the cells to a more continuous environment?
No, I will say it has no impact, but we are planning tests to compare for sure. It's really important. So we are currently doing tests to compare to the benchmark. We are working with clients that want to compare with their benchmark, and later on we will compare also. Does it have an impact on the production?
And I would even argue, even if it's different, I mean, if you're developing a new product, I mean, if different is better, assuming that perfusion is better, you could actually then develop the process like this and run your N stage in a fed batch mode.
Yeah, typically that would be the goal.
Yeah. Okay. Now let's look at the sampling because that's another challenge we outlined before. How are you tackling the whole sampling and the whole, I'd say, monitoring of the whole cell culture process?
So our system is really similar to typical rocking motion bioreactor with sensor. So we include pH, dissolved oxygen, and also a capacitance analyzer. In parallel, you can still connect an automatic sampler or take manually samples. But really, our vision is to be able to do the whole cell expansion without touching the system because you have the sensor inside, you can control the cells. So ideally, you just inoculate and then everything is automatic. But obviously, we want to check at some points if the pH and the dissolved oxygen and even if the cells concentration is the correct one. So typically, I think our client will take 1 or 2 samples just to check, but it will be just a way to verify if the measurements are correct. But if there is a drift of the pH, typically, anything else, that would be just control. It will be just verification, not control of the process.
Which potentially could mean that you can reduce the number of samples you draw because you manage very well the entire process.
Yeah, if you don't have any, if the dissolved oxygen, pH, and grow is going in the direction you have been seen in the last few runs, you can also adapt and reduce the number of samples because you have all those data that you were not able to get before when it was in check flask or spin tube because there is no sensor integrated. So now you can really have more control about the system.
Yeah, I think if we look into the future, it could also enable some sophisticated PAT technologies because you have the sensors already in your system.
Yeah, for version X in the future, we are going to integrate Raman or more advanced sensor. But for today, as it's a cell expansion, we focus on the more important one, so pH, dissolved oxygen, and cell density.
Yeah, I agree. I think you don't need to make it complicated at this stage. I think if you simplify the operations already and reduce the risk of contamination, you have already made quite a step. And yes, I mean, if you then can add some other more continuous analytics, that's great.
Yeah, and then it can be used as a bioreactor typically, because now more and more people are using Raman in their bioreactors. So typically later it can be used for material generation, but even now it's already compatible with those kind of application.
And that wraps up part 1 of our fascinating discussion with François Carruzzo about reimagining the seed train operations. Join us next time as we explore the role of automation, predictive analytics, and regulatory considerations in this groundbreaking technology. Before we go, are you feeling overwhelmed by your CMC development challenges? Let's cut through the complexity together. I'm offering a free call to evaluate and refine your CMC development approach with precision. Visit BrullmanConsulting.com to book your session. Until next time, keep innovating. All right, smart scientists. That's all for today on the Smart Biotech Scientist podcast. Thank you for tuning in and joining us on your journey to bioprocess mastery. If you enjoyed this episode, please leave a review on Apple Podcasts or your favorite podcast platform. By doing so, we can empower more scientists like you. For additional bioprocessing tips, visit us at smartbiotechscientist.com. Stay tuned for more inspiring biotech insights in our next episode. Until then, let's continue to smarten up biotech.